for the last session, and I'm going to breeze through my intro here a small bit, because I really want to get onto the results and have a bit of discussion and see what you all think about it. So, biogeography, marine tropics, we'll talk about factors that have shaped the evolution of fish and coral reefs. And to start off, I'm going to talk about something that comes up on my slide. It's the forward button. Okay, I'm talking about the Indo-Australian archipelago marine biodiversity hotspot, and this is different from terrestrial biodiversity hotspots because here centres of marine endemism do not equal centres of biodiversity. All right, and we still don't know how patterns of endemism and what the role of biodiversity is. Also, we have both latitudinal and longitudinal gradients of biodiversity, and we're still trying to figure out what processes drive latitudinal and longitudinal patterns. Thirdly, this is what I most want to talk about today, is tropical provinciality. So we've a habit of splitting up the tropics into realms, regions, provinces, and ecoregions. But I really want to try and understand, well, has this changed through time, and what kind of temporal patterns of tropical provinciality are there?
Maybe you could try pointing.
So I'll have to think.
Hang on, toggle for it.
All right, thanks. So anyway, right, starting off with species richness and endemism, you can see quite easily here that, so this is the distribution of species ranges from this data set by the Gaspar group. So it's over 6,300 species of fish that live in marine coral reefs, and so this is their distribution. You can see up top, species richness, you have that bullseye in the center, which is the Indo-Australian archipelago. But down here, if you look at the distribution of endemic richness, you can see that it doesn't match the same pattern. On the periphery, you have more endemics. So endemics have been important for trying to understand in this system how does this pattern come about. So often people talk about centers for speciation, or places where lineages have arisen, and often centers for endemism have been called centers for origination. But it's not quite that easy. So if you think about endemism, there are two types of endemics that you can have. One's a paleo-endemic, one's a neo-endemic. Paleo means old, neo means young. So basically we can have old endemics, we can have young endemics, and this is a little plot that kind of describes the processes of paleo-endemism versus neo-endemism. So paleo-endemism is mostly driven by extinction, so things are just about to go extinct, so where they are doesn't necessarily reflect where they've originated. Whereas neo-endemics is you're assuming that where the species is where the species arose. But you can also have things that don't start out as an endemic, so if you think of vicarious events, where a lineage might never have been an endemic before. So it seems important to try and understand at least the temporal pattern of endemism through time. So I had this idea to go, okay, well, we have all these big trees, and all the work I'm going to show now is probably obsolete because we've got even bigger trees on the way. But basically I want to say, okay, we should be able to get a good grasp of the age of endemism in this system, and maybe have a look at those sort of patterns. And so looking at this big data set, I identified 600 endemic taxa, but I can only get age estimates for 10% of them. So this is the pattern, but we can't even be sure if it's a pattern. But anyway, we've got old endemic lineages, we've got young endemic lineages, and if I put these up on a map, they'd be everywhere. They're inside the IAA, they're outside the IAA. So the first part of my talk is pretty bad because I have no answer to that first question. We still don't know what the impact of endemics on characterizing biodiversity in the Indo-Pacific. So I think this is a really nice kind of place for us to go in the future, especially with sampling more, especially more endemics. We might be able to get a better idea of this. Okay, so just to highlight quickly the work of Alexandre Cicada, and I just mangled his name, sorry. So he gave a lightning talk on some work we did in the origins, latitudinal biodiversity gradient in fish, and just to briefly say that basically the overall pattern is that we have higher net diversification rates in the tropics than in extratropical regions. And this is actually in contrast to what Michael Farrell said the other day. So, yeah, and if you want to ask me why that is, you have to wait till the end. Okay, moving on. And so, okay, we want to go the other direction, okay? Look at if the Centre for Biodiversity, or if the Indo-Australian archipelago has higher species richness because it's a centre for origination. So, is the hotspot actually hot? And this is work I'm doing with Michael Farrell and student Mary Shen. And again, we use this GOSI state-dependent speciation extinction. So if you're upstairs today, there's some things that aren't quite right with that. But anyway, the pattern here is that there is no pattern. There's no higher speciation when you look at these cross-section of these groups. Basically, there's no pattern of consistently high speciation rates inside the IAA versus outside. But there is a pattern in some groups where you might have higher dispersal rates going into the IAA rather than outside. And this kind of, when I first looked at this, I was like, what the fuck? No, this makes sense. Alright? Because back in 2013, I published a paper that said, oh, look at all this massive origination going on in the IAA. You know, it may have higher rates of diversification. Okay, so just to explain this plot, I lovingly call this the funnel figure. So basically, I split up, from an ancestral biogeographic reconstruction for the labyrinths, I split up all their nodes into and lineages into basically what I consider the four main processes driving biodiversity. So we have accumulation. So in the Paleocene, you see that accumulation seems to be a strong process going on. And what accumulation is that there's lineages there in the IAA that they're widespread. You can't say they didn't originate in the IAA, but they're there. All right. And then in the Oligocene, we have what's called survival, where we have lineages that are just hanging around in the IAA. They're not present anywhere else. They're just staying there, surviving. And then we have origination, where those lineages are actually giving rise to new lineages, and then some of those lineages stay constant or they further diversify. And then, of course, we've got export. So this kind of doesn't make sense when you look at the previous figure. But it actually makes a bit more sense when we do this for every other basin. So same color code before, all right? Where you can see instantly, first of all, the Indian Ocean and Central Pacific Islands, they get most of their diversity from the IAA. Okay, so all of their stuff is accumulation. But there's still quite a bit of origination going on in the Atlantic and some in the East Pacific as well. So what the whole pattern of not finding higher rates of diversification or higher rates of speciation in the Indo-Pacific or in the IAA could be linked to this fact that there was more lineages there, but they were just diversifying at the same rate as everywhere else. Okay, so it seems to be more important that the survival of ancestral lineages in that Oligocene period seems to have driven diversification in the IAA and the pattern we see today. Okay. Okay. I'm going to take a little break now. So this is kind of the core of what I want to talk about here. So tropical provinciality. We've split up the tropics in a number of different schemes over the years. But I want to draw your attention to the most recent one by Kubicki et al. In 2013. So this, again, is based on that Gaspard data set. This group of international collaborators who got together to put the geographic ranges of over 6,300 species in a data set and do some really cool analysis with it. So this is an assemblage dissimilarity estimates for all of these geographic locations, and basically what it's showing is that they're clumping regions together that are more that have similar assemblages. So this is just based on species presence and absence in all these regions. So again, when I saw this, I started thinking, okay, well, what does this mean for the evolution of the system? Do any of these geographic schemes make any sense on an evolutionary timeline? And any previous biogeographic analysis I've done, I've always said, okay, this is the Indo-Australian archipelago, these things are here, this is the Indian Ocean, they're there, this is the tropical Asia Pacific, these things are there, and then you have the Atlantic, okay? But how about we let the data speak for itself and see if we can actually recover any sort of tropical provinciality from the data set. So, move forward. So, I grabbed as many phylogenies as I can find for all these groups of iconic reef fish, and then threw it onto a larger, kind of cobbled together a big tree. And I used this program called Bay Area, which is Bayesian Biogeographic Inference by Landis et al. And so this basically gets over the problem with having too few areas when you're using Lagrange or BioGeoVaries. So basically this allowed me to do ancestral range estimation for across 111 ecoregions. Okay, so this is using the Spalding tropical ecoregion designated. So this is they're relatively small areas that are defined on environmental similarity and a couple other characteristics. So for this, every point on the tree becomes a location or a lineage. I can get basically the presence absence of that lineage across these 111 ecoregions. So I'm going to get a sense of lineage dissimilarity through time and hopefully be able to look at temporal structure in provinciality. Okay, so that thing's saying, yeah. So this is what we get for present day. So this, let's get the. So this here is our phylogeny I use. So we've got all the black nodes there. What this is looking at the relationship between all of those little ecoregions there. So this is a hierarchical classification of dissimilarity among species assemblages. following the correct and yes method. Okay, so this is from the Berdell study in 2012. So I'm getting a sense of phylogenetically informed assemblage dissimilarity. And so we can actually directly compare this with the Kubicki et al. Study. Okay, so the Kubicki et al. Study doesn't have a phylogeny behind it, but it's basically from the same data. It's just a subset of it. And additionally, the first difference that you can see is that the Central Indo-Pacific area that's right down here, this actually engulfs the Central Pacific and some of the Indian Ocean as well. But something weird seems to be happening. You've got a little green thing up, green blob up here, and it seems to be so. This is some of the Japanese islands is linked with Easter Island over here. So basically, I was kind of stumped by this. I didn't really know why it would put these two things together. But then I remember it's taking phylogenetic similarity into account. So when you look at the species that are driving this pattern, it's actually things, they're little endemic species that are budding off from the same clade. So phylogenetically, they're close. But these are, it's really pointing out centers of endemism here. Next. And then we also, so that there's a basal split between the Indo-Pacific and the Atlantic here. But notice, I didn't show it the last slide, but the tropical East Pacific was more closely linked